drug discovery process and development process. Then we will go to different stages of drug development. There are five stages which I'll talk about. And finally, I just quickly will touch base on, what is happening on AI side in drug development. There's a huge development in that, and people should be aware of what how it is helping the drug development process. And finally, we'll have summary and questions. So uh, let me start asking these questions. I know uh, when you start asking questions, then people get to know where we are going. So, what are the objectives? Why any company would like to develop a new drug? It's not just because they have a lot of money, or. So there are clearly three reasons why a new drug would come into market. One is First thing is to have a higher efficacy than the existing drug. So if you want to have a higher efficacy, that's the primary thing. Or you need to you don't have anything for a particular target, then you start developing. So you need that efficacy for that new target. That's the first objective. That's where you start saying, oh, I want a new drug. Second is the current drugs in the market might not be as safe as expected. So you want to come up with some drug which would be higher in safety. It would be more safer than the existing compounds. So that is the second objective. And then third is maybe you want to develop something which is of lower cost. So these are the primary three objectives why any company would like to come up with new drug. So efficacy, safety, and cost. Efficacy might be as compared to the current drugs, or you're targeting something new. So now that's in nutshell why anyone would like to have a drug discovery or drug development process. Now, when we see, is it really a you know how how easy or how difficult it is. So correct, it's really a very risky process, I would say, uh, because if you start, and this number might change, some might say 5,000 or 10,000, but it's in thousands. If when you start with thousands of drugs, or drug molecule candidates, finally you land up with one drug molecule. So there's so much of attrition because you want the drug to be, it should be potent, it should be selective, it should be targeted, it should be long lasting, it should be non toxic, and it should be patentable, manufacturable, druggable, so you need to have all these characters in a new compound, but that's not possible, so that's where when you start with, maybe 10,000 candidates, you'll end up with one drug molecule being there. That's such a less amount of percentage of success there. So it's, I would say, drug discovery and drug development, yes, it is science, but it is more of an art also. What do you do and what you don't do. So now the second thing is it's very time consuming today. It takes at least minimum 12 to 15 years to develop any drug. Uh, right. So if you really see, I will go through this process. As I said, there are five main stages of drug development. One is first is discovery, then comes the development piece where you have got preclinical and clinical phases. Then there is a FDA review, and then there is a launch. So these are five stages. And if you see the first discovery and preclinical stage itself, it will take 3 to 6 years. The clinical phase would take again 6 to 7 years, and add again, maybe 1 to 2 years for FDA approval and answering their queries. So, and that's again, when you see 5 to 10,000 compounds, how many are dropped during discovery? Almost, only maybe 2 to 300 would come to preclinical phase. After preclinical phase, there are only five candidates which remain to go into clinical trials. And after clinical trials, they fail there, and then finally one drug comes to the market. So that's a huge attrition there. But at the same time, you are spending 10 to 15 years. Now, you can just imagine how much of an effort it is there to pick a right candidate, because this all requires time, money, efforts, resources, and no company would spend resources and money just for the heck of it. So, and that's why I say it's more of a science, but it's more of an art. It's an art also, what to do, what to select, How do you go ahead? That's where your skill comes into picture. Now, why this attrition? Why this? Why I continuously say it's an art? Because if you see reasons for failure in the preclinical phase, 44%, almost half of the candidates, they fail because of toxicology. They become toxic there, and you say, oh, I need to abandon this. When you go to phase 1, the clinical safety is again around 25 to 30%, where the candidates get dropped. So if you look at either clinical safety or preclinical safety, almost maybe 70% of the candidates drop here. And when it comes to phase 2, it's the most of it is because of the efficacy. Whatever you have learned in preclinical stage doesn't translate into human kind of efficacy. So 50% candidates drop here, and again 72% drop. This is in these ways, how many come and how many are dropped out. It is not the total number of percentage what I'm talking about. Otherwise, it would go above 100%. So let's look at it. Drug development is a team sport. Why I say so is, first of all, it's not a one man job or one team job. It's a huge effort coming from different faculties. you know First is, and that piece is missing here, I'm sorry for it. The first piece is medicinal chemistry and analytical. They are the ones who start developing libraries. Then it goes to formulation scientists. Can I formulate it? Whether it is soluble, whether it is, how can I formulate to start using it? Then comes, a stage where toxicologist comes in picture. Like, how is safe? How do I perform the toxicology studies? And then when it goes to clinical operations, it is, clinical pharmacologists, safety physicians, clinical development. How do I monitor these trials? How do I design the trials? How do I conduct the operation, clinical operations? Those are clinical people come in picture. And finally, once all this is done, it goes to the regulatory people, because they know the regulations, what information is required, how to put it, how to put the dossiers. They would come into picture and start developing the dossiers for final submission. So it's all the teams coming together, and then looking at the drug development piece. So that was just a quick background about how what is the background about drug development process. Now we'll start with really what are the stages of drug development. The first stage, and there are 5 stages as I said. The first stage talks about drug discovery and lead identification. What is lead? Lead is your lead candidate. So so as I said, there might be thousands of molecules which are which are suggested by the medicinal chemistry people. They will look at chemistries and start developing molecules. Now, out of that, which one is to be taken? That the the candidate which goes forward is called as lead candidate. So the first stage is drug discovery and lead identification. Once the lead is identified, you take it to the preclinical stage. The preclinical studies, toxicology studies, pharmacology studies, which are called as pre-IND studies. We'll go into that later. And then once you have this, so this is all about animal studies, because no one can go to human beings directly. First, you have to test in animals and then go to human beings, obviously. So once you have that animal data, you submit something called as IND. IND is investigational new drug to the regulatory authorities. They look at the data, they say, okay, fine, you have some candidate you can go to, and it's safe you in animals. Now you can go to clinical trials. So then the clinical trials starts. Clinical trials have got 4 stages: phase 1, phase 2, phase 3, phase 4. The phase 4 is post marketing. Why I have highlighted in red, I'll come to that. Once you have this phase 1, phase 2, phase 3 data, then you submit all that data, preclinical data plus this clinical data, for to the regulatory, which is called as the NDA. NDA is new drug application, and then they go through the review process and approve it. Once they get to approve, this product is launched. Now, after launch, your process doesn't end there. There's a phase called as post marketing, and this coincides with your phase 4 also, so this phase 4 of clinical trials, and this is almost the same, so that is why I've highlighted in red. This post marketing would look into, what are the events, adverse events once it is launched. There are few products which have been withdrawn from the market because after continuous use in the market, they have seen severe adverse effects, and that's why the drugs were withdrawn. So that's where the pharmacovigilance piece comes into picture, and post-marketing vigilance is that fifth phase. So that is how the stages of drug development are divided. We'll go one by one into each one. So this is the map. Let me first talk about when we say drug discovery and drug development, what is the difference? So this is discovery, this is development, and this is post-marketing. Here it is, you have the launch. Now the discovery phase is all about, you are playing with the candidates. It's all exploratory. You know, you try to, see which one is good, which one is bad, which one is not, which one is best. And for this, you do some exploratory studies, which is called as the discovery studies, wherein you play with all the candidates and then go to lead identification and validation. And finally, you select the candidate. Once you select the candidate, then the drug development starts. What is the difference? Here you conduct all the studies which are meant for submission. That means there's no room for exploration. There's no room for, you have to do studies as per regulatory norms, as per GLP norms, and what is, we'll come to that, what, why we need GLP studies. So these all studies are conducted, maybe preclinical or clinical, as per regulatory requirement, as per regulatory norms, as per regulatory guidances, and then only they get accepted for, IND submission or NDA submission, and they go for FDA approval. And then there, if it's okay. So you have got discovery phase, you have got development phase. Once it is launched, it goes to the market, and then you have the post marketing or post launch, you know, um, kind of observations. Now, why, like, when I say this development is as per regulatory norms. So in the world, there are many guidelines which are available globally. So US FDA guidances are, you know, followed mostly everywhere. And then there is European medical agency. So you have got FDA guidances, you have got your EMA guidances, and you have got Japanese guidances also. But then there was few differences within these guidances. So they all came together and formed something called as ICH, International Committee of Harmonization. So ICH has come, they have taken all the pieces from these three major guidances, and they have formulated ICH guidances. Now, so worldwide, ICH is the one which derives or which drives this, what studies are required, how they should be conducted, for what duration they should be conducted, you know what are the, what are the situations or norms they need to follow. Otherwise, if you don't do according to it, the FDA would or the regulatory authorities would reject your proposals. So that's where the discovery phase is important, the development phase is important, to see how your drug can get approved as soon as possible. Now, this is the money piece or money slide of what you said. This is the core slide where I'll spend maximum amount of time. So if you see, as I said, this is a map of non-clinical studies, when the toxicology studies. I'll come to the clinical phase later on, but this is the preclinical phase. And so this is the exploratory, the drug discovery phase, and this is the regulatory or development phase, the drug development phase. Now, for any studies to be started, as I said, first would be your drug discovery or exploratory studies. Now, when I say exploratory studies, you will see that you have different kind of studies which could be done. As I said, exploratory studies, you can play with the molecule. So the process starts with medicinal chemist. They have library of chemistries or chemicals they will produce. Now you cannot take all of them. So one thing is, okay, the company says, I want to develop a drug against diabetes. I want to develop a drug against Alzheimer's. I want to develop a, so they will look at, all the information, what type of chemistry is required, what type of, chemical structures are required, so that the molecule can penetrate, or go to that site. So they would look at it and start producing or synthesizing chemicals. And that's where all these studies would be required, you know, for drug discovery. Now, the discovery studies, this starts with literature, sir. So once the company says, we want to develop a drug for Alzheimer's, so now the chemist would know that, okay, these are the existing drugs against Alzheimer's. They have got this kind of structures. They have got this kind of structures which would be safe. This kind of structures would be, I'm talking of chemical structures. Chemical structures which might be toxic. These are the structures which won't enter brain. These are the structures which might enter brain, would pass the brain, blood-brain barrier, and so on. So for each target, you take of anything, kidney, heart, lung, or brain, any target, they would have to start with a literature survey. What is known about, the current molecule? So that is the very first stage, literature survey. This is not only done by medicinal chemists or chemistry people. It is done by toxicologists. It is done by clinical pharmacologists. It is done by regulatory people. Whatever is existing is first should be known, and then you can modify it. Or that could be a cue where we should go, where we should not go. So first, after literature survey, then comes something called as in silico prediction. Now, if you talk of in silico prediction can be done for toxicology. Although I am talking of toxicology now, it can be done for toxicology. It can be done for medicine chemistry. It can be done for pharmacology. It can be done for anything. Now, what is this in silico prediction? In silico prediction is something called as, you have got computational models, and that's where the AI in toxicology or drug development started. What was done was, if I don't know whether you have heard these words, Derek. These are, all, uh, what do you say, um, tools you have today, which have been accepted. There are few tools which have been accepted by the regulatory authorities also. So there is Derek, there is MCase, multicase. These are the models which are accept, or tools which are accepted by FDA, EMA, and worldwide. That means whatever data you generate through this is accepted. You don't need to conduct the studies. So how these models have been developed? These models have been developed by. So it all started with, developing a tool for, say, cancer. I want to know whether my drug is going to cause cancer or not. So what they looked at is, they collected information on all the carcinogenic chemicals, looked at the structures, you know, looked at the literature, put it in the, machine learning kind of thing. That's where machine learning started. And then once you put your new structure, the machine would learn, okay, this structure is, you know, close to this structure, which is cause cancer. So they are going to give predictions. So this will give you the predictions. The models I have mentioned here is, for toxicity. One is direct, one is Topcat, other is multi-case, and there are multiple models. If OECD toolbox has come, there are few which are paid, which are a few which are online available. They are free of cost. You can put your new structure there and see what is going to be the prediction. So these are predictive models. So these models which I have written here, they would predict, mutagenicity, whether your molecule is going to be mutagenic, carcinogenic, reproductive toxic. They would tell, for kinetics, they would say whether it is going to absorb, it is going to be metabolized, whether it is how fast it is going to be excreted. So you've got models for pharmacokinetics, you have got models for pharmacology, you have got models for, predicting what are the side effects. So there are different models which are available. This is just a glimpse of, what are models which are, available for toxicology. And in this case, I've just given you two examples. There are many which are available today. One is, rule based, expert based or rule based. Means is structured based. Like you have put different structures, and then they will match with the structure and say, okay, this is structure based. The other is knowledge based. What is the literature saying, what is the knowledge behind this, and then the structure based and knowledge based would come together and form a, a model. So Derek has got both the components. Few of them have got only, the this top cut model is QSR model. It is based just on structure. So when FDA has gone through these different models, they have undergone the validation process, and then they have accepted, as I said, two or three tools, which where you can put your models and say, okay, fine, this is not going to be mutagen. And once this predicts it, FDA will say, I don't require any study. This model is fine with me. So you can avoid studies, you can avoid time there, and then you can test in a day. You can put hundreds of compounds and say, okay, this 95 are going to be mutagenic. I'm going to pick only five of this. So that's where your lead optimization starts. So one first was literature survey, second was in silico models, the third is in vitro studies. These are all in vitro studies which are cell based assays, tissue based assays. And you will again see, so for discovery studies, you want to know your molecules, whether they are going to be causing hepatotoxicity, whether they are going to cause, toxic to blood cells, whether they are going to cause muscles, toxic to muscles, whether they are going to be genotoxic, whether they are going to cause any chromosomal damage, they are going to cause Purkinje fibers, are they going to be cardiotoxic? So you have got cell lines for different organs, and then you study your molecules. So first is literature survey, second is in silico. So you start filtering. So maybe your chemist has given you thousands of molecules. Now you start filtering. Okay, the first 50 based on literature survey, not good. Second is based on in silico, next 25, not good. So then you lay whatever remaining, you start filtering and then see, okay, then you start conducting in vitro study. Now, if you see, this all process is going from, to reduce the timeline or reduce the timelines. You have got hundreds of molecules, you're just filtering based on quick studies. So these in vitro studies, they are comparatively, as compared to in vivo studies, they would be quick, they would be, mm, less costly, and you can get an idea what your drug could cause or not cause. So similarly, you know you can do all these studies and say, okay, fine, after all these studies, I have come up with, maybe 15 candidates now, which clear and which doesn't have any in vitro toxicity, or they are good in vitro pharmacology models also. Mind you, this is, you have to study everything. You have to study kinetics, you have to study pharmacology, you have to study toxicology through these in vitro studies. And then once you filter out, okay, in vitro studies, these things are good, these things are not good, then you shift to in vivo studies. Now in vivo studies, you have got, again, being discovery, it's exploratory. You don't you use huge number of animals. Now, from management perspective, management will always say, use as less number of studies as possible, use as less number of animals as possible, because it costs, there's money involved, and they would, management would try to reduce the cost, whereas regulatory would say, give me more data. So there's always a clash between what management says and what regulatory says. So the toxicologist, the pharmacologist, the clinical people, they always need to balance that, demand, from regulation versus what management is doing. So in exploratory stage, it's just to